First off is the monohybrid cross. In pea plants, spherical seeds, S, are dominant to dented seeds, S. In a genetic cross of two plants that are heterozygous for the seed shape trait, what fraction of the offspring should have spherical seeds? So first off, we need to draw a Punnett square. These will help determine the possible outcomes of both genetic and physical traits. Now that we have one, like so, we need to determine the genotype of both parents. The problem states that both plants are heterozygous, so both will be capital S, lowercase s. One dominant, the big S, and one recessive, the little s. Since there is a dominant allele in both, the two parents are both spherical seeds. However, when we put the two parents on the Punnett square, something surprising happens, which we'll get to in a little bit. First, we separate the two letters and put them each on the square like so. Now, the rest is actually quite easy. Just like multiplication, we can combine the two letters for each square. The two dominant S's make a homozygous dominant SS. Dominant and recessive make a heterozygous SS. And the same with the other square over here. Now, we have two recessive S's, and when we combine them, we get a homozygous recessive SS. Now we have a complete monohybrid cross, but what does all this mean? Well, looking at the square, we have one homozygous dominant, two heterozygouses, and one homozygous recessive. This is where it gets interesting. Since the first three have a dominant allele, they will be spherical seeds. This is the answer to the problem, 75%. But even though both parents are also spherical seeds, we have one group of dented seeds. However, the outcomes of Punnett squares depend on the genotypes of the parents, so this will not always be the case. That's monohybrid crosses, so now it's time for dihybrid crosses. In pea plants, spherical seeds, S, are dominant to dented seeds, S. In addition to this, tallness, T, is dominant to shortness. In a cross of two pea plants that are heterozygous for both traits, determine the phenotypic ratio of the offspring. Now, this will be a little bit more complicated than the monohybrid cross, but they follow most of the same concepts. First, we draw a 4x4 Punnett square. This is because there are 4 letters, not 2. Then we fill in the top and side with all possible combinations of the two traits for each parent, like so. Since both parents are heterozygous for both traits, they both have the genotype SSTT. Just like the last time, we fill in the inside with the combination of the two parents. Done. Now we count the different possible appearances of the offspring. These 9 will be spherical seeds with tall plants. These 3 will be spherical seeds with short plants. These 3 will be dented seeds with tall plants, and the one that's left will be dented seeds with short plants. Therefore, our phenotypic ratio is 9 to 3 to 3 to 1. Next is pedigrees. So, as far as a problem for this one, I'll put up an incomplete pedigree right here. So, the use of a pedigree is to follow a trait as it is passed down from generation to generation, and to determine the genotype of different family members. First off, in the key are symbols for different parts of the pedigree. The trait is recessive. As you can see, Kevin and Martha have 2 sons and a daughter. The first son, let's name him Jeff, is unaffected. He marries Louisa, and they have 1 son and 2 daughters. Only the one daughter is affected, so she is guaranteed to be homozygous recessive. Let's use S for genotypes. So she is SS. Now, while we don't know the genotype of the other children, we can tell the genotype of the parents. Both are heterozygous, or else they wouldn't be able to have an affected child. As far as their other children, we know they have at least one dominant allele, but we don't know both. We can put a question mark, as we won't always be able to know the genotype of everyone. On to Kevin's other son, George. He has the trait, so he is homozygous recessive. Their daughter, Molly, marries a man who is affected by the trait, homozygous recessive. Somehow, all 5 of their children are unaffected. While she could be heterozygous, that's most likely not the case. She's probably homozygous dominant, meaning all of her kids are heterozygous. As far as the original parents, we know they have a child who's affected, meaning both of them must be heterozygous. And that is a pedigree. Now on to sex-linked genetics. As usual, there's some background information you'll need to know beforehand. First off, females have 2 X alleles, and males have an X and a Y. Now, certain traits are linked to a certain allele, usually the X. Let's talk about cats. Say there's an X-linked recessive disorder called 8 lives disorder. We call it E. A female cat is heterozygous for this disorder, and their genotype would be XE. She falls in love with the perfect male cat, one who is dominant on the X allele. What type of offspring can they have? Start by drawing a Punnett square. Now, we know the genotype of both cats, so fill in the outside like so. The male cat has the genotype XEY. Then just fill in the inside like a normal Punnett square. Now, both of the female offspring have a dominant allele, which they get from the father. So no female offspring will have this disorder. As far as the male cats go, they get their X allele from their mother, which means there's a 50-50 chance they get the disorder. Next, we'll discuss codominance and incomplete dominance. Say we take the Punnett square we made in the last problem, except this time the trait is color. Instead of having a dominant and recessive allele, there are 2 separate alleles, O for orange and B for black. Fill in the Punnett square like so. The mother is orange on both alleles, and the father is black. The result of the Punnett square would look like this. In the case of codominance, both the O and B are dominant and are both expressed, just separately. So all female cats would be XB/XO, or a calico cat. No male cats would be calico since they don't have 2 X alleles. In the case of incomplete dominance, neither trait would be dominant over one another, so they would combine. A black and orange allele would make a dark orange or brown cat. Not all traits are like this, however, so keep that in mind. Next is blood type genetics. A man rushes into a hospital, losing blood rapidly. The doctors keep him from losing any more blood, but he loses enough that he needs some more. However, using the wrong type of blood can be catastrophic. So why is this, and how do doctors make sure they get it right? First off, there are 4 main types of blood: A, B, AB, and O. Each type is based on the types of protein in the blood. Type A has A proteins, type B has B proteins, type AB has both A and B proteins, and type O doesn't have either. When somebody receives blood with a certain protein they don't have, clotting occurs, which is not good. However, a person can receive blood that is missing proteins. An example is that a person with type A blood cannot receive type AB blood, yet a person with AB blood can receive type A. This means that type O blood is the universal donor since it doesn't have either protein. So in this scenario, the doctors are most likely to give him type O blood, since they can guarantee that it will be compatible. In less urgent scenarios, blood can be taken from the patient and combined with other types of blood. Their reactions to the other blood types will determine the patient's type of blood. Then, a type of blood other than O could be used accordingly. And that's blood type genetics. That's all for today. Thanks for watching.